Amino acids are the basic building blocks of proteins, and they serve as the nitrogenous backbones for compounds like neurotransmitters and hormones. In chemistry, an amino acid is an organic compound that contains both an amino and carboxylic acid functional group, hence the name amino acid. Proteins are long chains or polymers of a specific type of amino acid known as an alpha amino acid. Alpha amino acids are unique because the amino and carboxylic acid functional groups are separated by only one carbon atom, which is usually a chiral carbon. In this article, we will solely focus on the alpha amino acids that make up proteins. Proteins are chains of amino acids that assemble via amide bonds known as peptide linkages. The difference in the side chain group or R group is what determines the unique properties of each amino acid. The uniqueness of different proteins is then determined by which amino acids it contains, how these amino acids are arranged in a chain, and further complex interactions the chain makes with itself and the environment. These polymers of amino acids are capable of producing the diversity seen in life. There are approximately 20,000 unique protein-encoding genes responsible for more than 100,000 unique proteins in the human body. Although there are hundreds of amino acids found in nature, only about 20 amino acids are needed to make all the proteins found in the human body and most other forms of life. These 20 amino acids are all L-isomer, alpha amino acids. All of them, except for glycine, contain a chiral alpha carbon. And all these amino acids are L-isomers with an R-absolute configuration except for glycine, no chiral center, and cysteine, S-absolute configuration, because of the sulfur-containing R group. It bears mentioning that the amino acids selenocysteine and pyrrolysine are considered the 21st and 22nd amino acids, respectively. They are more recently discovered amino acids that may become incorporated into protein chains during ribosomal protein synthesis. Pyrrolysine has functionality in life. However, humans do not use pyrrolysine in protein synthesis. Once translated, these 22 amino acids may also be modified via a post-translational modification to add further diversity in generating proteins. The 20 to 22 amino acids that comprise proteins include alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, valine, selenocysteine, pyrrolysine, not used in human protein synthesis. Of these 20 amino acids, 9 amino acids are essential: phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, leucine, lysine. The non-essential, also known as dispensable amino acids, can be excluded from a diet. The human body can synthesize these amino acids using only the essential amino acids. For most physiological states in a healthy adult, the above 9 amino acids are the only essential amino acids. However, amino acids like arginine and histidine may be considered conditionally essential because the body cannot synthesize them in sufficient quantities during certain physiological periods of growth, including pregnancy, adolescent growth, or recovery from trauma. Although there are 20 amino acids required for human protein synthesis, humans can only synthesize about half of these required building blocks. Humans and other mammals only have the genetic material required to synthesize the enzymes found in the biosynthesis pathways for non-essential amino acids. There is likely an evolutionary advantage behind removing the long pathways required to synthesize essential amino acids from scratch. By losing the genetic material required to synthesize these amino acids and relying on the environment to provide these building blocks, these organisms can reduce energy expenditure, especially while replicating the genetic material. This situation provides a survival advantage. However, it also creates a dependency on other organisms for the essential materials needed for protein synthesis.